Thank you. I would like to thank the organizers and all the speakers and the attendees for this wonderful meeting. So we humans have developed the theory of quantum electrodynamics, and we can predict the properties of electrons to more than 10 digits. We have landed a man on the moon, and we have built machines that can demolish our world championships in games that require high-level human reasoning. But we still do not understand how we can recognize and discriminate between these dogs and muffins, and we do not have computer algorithms or computer vision algorithms that can easily solve this problem. And despite tremendous progress in the last 60 years or so in trying to understand how the visual system processes information, we still do not understand algorithmically or have a circuit-level mechanistic understanding of how complex visual inference problems are solved in any mammalian species, so how this problem gets solved. And at the heart of trying to understand the problem at the algorithmic level is to understand representations. How is information represented and transformed along what we now know as the visual hierarchy? And there has been tremendous progress over the last years, but we still do not have this algorithmic understanding, and this is how I want to begin my talk. But before I do so, I would like to acknowledge the people in my lab, my collaborators, and I will be going through them during the talk, the people that contributed, and of course our funding agencies. So representations and transformations lie at the heart of trying to link the patterns of photons that arrive on the retina to behavior. And there's been a lot of breakthroughs, and I just here list some of the most fundamental breakthroughs, and a lot of them led even to Nobel Prizes in the last, since the 40s. Starting here with Hardline, Hubel and Wiesel, the work on the retina by Barlow, place fields discoveries, Charlie Gross, and the grid cells. But a lot of these discoveries in trying to understand representations was a combination of serendipity and intuition in science. They were not, a lot of these discoveries, the scientists that did them, they didn't go there trying to have a hypothesis. And there's a very famous example of this, of Hubel and Wiesel, how they discovered orientation selectivity, which was an accidental discovery. They were using spots of light to try and figure out how the retina, how V1 neurons respond. And one day there was some dirt, the experiment wasn't perfect, and they noticed that it was the edge of the slide that was causing the effect. And if you look at the brain, I think I was one slide behind, but if you look at the brain, there's a lot of visual areas in the macaque, for instance, and in many other species. There's many layers, many different cell types, and we do not have even, at the level of individual neurons, and it gets even more maybe complicated at the population level, an understanding of what information is represented. And the reason is that in vision, for example, the problem is high dimensional. If you have a monitor that you can generate, let's say 256 by 256 black and white pixels, you can generate 2 to the 65,000 patterns, and there's only 2 to the 300 electrons in the whole universe. And because it is a nonlinear transformation between the input and all these stages, the problem becomes almost intractable, and that's one of the reasons why a lot of these discoveries are very slow to come, and they are mostly happening accidentally or by serendipity or some deep intuition that these people have. So what I'll describe to you today is one attempt that we have done in the last few years in trying to solve this problem by developing a paradigm that we call Inception loops. And the experiments that we'll discuss today, we apply this both in mouse and the monkey visual system, and more recently in other sensory systems. And the experiments that we'll describe today were done by Talia Mohammed, a very talented experimentalist, and also the data analysis and machine learning by Edgar Walker and Fabian Sim. So Inception loops comprises of 4 steps, and I will take you through it and show you some data. The first step is that you have to collect a lot of data, and this is something that, as it was shown earlier today, this is one of the wonderful things that happened in the last 10 to 15 years. We now have technologies that enable us to record a lot of data in volumes. And in this case, we're going to use two-photon imaging to record neural activity using this mesoscope from the mouse visual cortex. And here we can record about 5,000 neurons simultaneously at 8 hertz, and we can show 5,000 natural images. So within a period of, let's say, 2 hours, we can collect in the order of 25 million training data points. The second step is because of this, again, quote-unquote revolution that happened in the last 10 years with deep learning, we can use deep learning as a system identification method to build accurate predictive models from this data. So we use neural network as a black box. We give it an input. We try and predict the output. And these data-driven deep learning models that can be applied to any system without even having prior understanding of what the system is trying to do. It's just learning a predictive model. Because of the large volume of data, as I told you, 25 million training samples, they're in par with, let's say, what Google and Facebook used to train their algorithms. We can generate very accurately predictive models. And these models, we've published a number of papers with our collaborators, including with Matthias Bethke here, who is in the audience. And they have different versions of them. Some of them, they take into account behavioral modulation too, like the pupil, eye movements, the rounding of the animal, even the decisions that it's making. And these models are extremely good. They perform, with different measurements as a fraction of oracle or some upper bound on how good they become. When you train them on a lot of data and many animals, you can get close to 90% of prediction with these, to arbitrarily predicting natural images, and we've extended them also to dynamics. So that's the second step. So the first, you collect a lot of data, then you build a predictive model. The third step is that these neural networks are not entirely a black box. You can try and open the black box and try and understand something. And the easiest way to think about it is that if you have a faithful in silico predictive model of the system you're trying to understand, you can do a gazillion number of in silico experiments that have taken, hundreds of years to perform in the animal in the first place, right? Because if you have enough money, you can just go to Amazon and you can just run this on many computers. You can also do something a little bit smarter. Because it's a differentiable model, you can start doing experiments that are almost impossible or impractical to do in the brain in the first place. For instance, you can go and ask the Hubel and Wiesel question, which is what they were trying to do when they admitted accidental discovery. What is the optimal stimuli that drive these neurons? Okay? And the way you do this is quite straightforward. Here you have this model. You start with some arbitrary input. It could be white noise. It could be a natural image. You pass the image through the network. You have a target neuron in silico now that models a neuron in the mouse's brain. And you try and optimize the stimulus. And you basically use gradient descent, but instead of changing the synaptic weights, you just change the input, the pixels. And you do this many times. You go back and forth. And this is actually a true optimization. And here it looks like we kind of rediscovered, if you want, the edge detector without following, our intuition as much or, accidentally discovering it. It was more, let's say, data-driven approach. So when we did this the first time a couple of years ago, we noticed that many of these most exciting images, or what we call MEIs, deviated from the standard notion of an edge detector or a Gabor. And this is primary visual cortex of the mouse. Many of them look like these weird type of shapes. My daughter, when she saw it, she said it reminds her of a Chihuahua dog. But the bottom line is that if I show you a bunch of them, and this is a representative sample of 150 of them, some of them look like what you would think maybe like a Gabor patch of different orientations, but many of them look like checkerboards, things that had of different orientations, sharp corners, black corners or white backgrounds, corners of different angles, curved strokes, and so on. Now, until now, it could all be an artifact of the model, right? There's a lot of assumptions that go into this modeling. The models have architectures that can induce, they're convolutional, they can induce different inductive biases, so all this stuff could be artifactual. But one great thing about it, now you can go into the step number 4. Once you've made what you think is an in silico discovery, you generate a hypothesis, because of the modern methods, you can go back and test it in those neurons in the first place. And here we do chronic recordings you know, these models typically take a whole day to train, and you do these experiments overnight. But we can go and find the same neurons and then test these ideas overnight. So we take these images, we show them back to these neurons, and here is the confusion matrix of the real responses. Each row here is the responses of one of these 150 neurons that I'm showing you here we tested, and to the responses to these 150 images. And the fact that you get this very strong diagonal in the middle is an indication that this model is synthesizing stimuli, and that's where the word inception comes from synthesis, incepting the stimuli. It gets the responses, it predicts them very well. And in fact, if you look at these models, they don't just predict the responses to these MEIs, but if you take all the MEIs and you now plot here, this is for one neuron, the predicted response from the model and the observed response, so now you take each row of this matrix, you see that you get a very strong correlation, and this is the distribution across this animal of many different neurons. Now, when we're trying to understand this, it's an ongoing process. What is, what scientific insights can we get further after we have done this optimization? And one thing that we've looked at is to see what do these most exciting images represent. And the way we did this is that we took a new set of thousands, 5 to 20,000 natural images and cropped them at the size of the receptive field, and now just filtered or passed the images through the model and found the most exciting novel images, the most exciting natural image. And when you look here, for example, on the left, the most exciting input that was generated by the model versus the most exciting image, you see that there is some perceptual resemblance between these most exciting inputs and naturally occurring patches in images. For example, here is like a corner. Here is something that looks like a checkerboard that happened to be part of a keyboard. This is like a crescent, a T-junction, and so on. So to summarize this part so far is that this approach enabled us to generate, to do a data-driven pipeline for discovering the representations, what is being represented in different parts of the brain. And what we found, and this is unpublished work, that in the macaque, when we do this in the macaque V1, we find that there's a much closer resemblance to the Gabor filter. So we find quite a remarkable difference between the primary visual cortex of the mouse and the macaque. But this method can be extended to many different parts of the brain. You can study whole tuning functions. You can extend it to populations of neurons and to dynamics. And this is what we are currently doing. So to summarize so far, these MEIs in mouse exhibit complex spatial features like checkerboards and corners. They deviate from the common notion that Gabor stimuli are optimal for V1. And there's this difference between the macaque and the mouse. And these MEIs, they occur at a certain frequency in natural images. And just to give you a number on that, let's say the level of sparseness, about 1 in every 2,000 images has a patch that will drive the neurons above 75% of the MEI. The MEI is always the best, so it gets to, so you can imagine that at least with calcium imaging in the primary visual cortex of the mouse, these are layer 2-3 pyramidal cells, you get this level of sparsity. So these are sort of tuned to natural patches, and they are occurring at that level of frequency. Now, what is interesting or exciting about this thing, this method of inception loops where you collect data, you build a highly predictive model, you do experiments in silico, all kinds of experiments to try and make discoveries and generate hypotheses, then you test them back. You can run this inception loop, of course, multiple times, and we're doing that. You don't have to. You can have, let's say, the model and the brain or the system you want to study looping continuously, in trying to optimize the problem. And you can apply this to many different questions, like you can, not just in neuroscience, but to any sort of difficult nonlinear optimization problem that you want to address. For example, you may want to create optimal stimulation patterns to, in Parkinson's disease or in depression that people are using. So you can try and, use in regimes where you can have the capability to collect a lot of data, and if you have enough data and you can build these predictive models, you can run this thing around. Now, the goal, though, the gap in knowledge, the way I described it at the beginning, was not just that we're trying to understand what is being represented, and we can march now with this method across the visual hierarchy in different layers, different cell types, extend this, as I said, to whole tuning functions to try and get some insights in how is information represented and transformed. But ultimately, being biologists, we're interested to understand mechanism. And to understand mechanism, we get inspiration here from Richard Feynman, who said, what I cannot create, I cannot understand. And I'll give you an analogy of what I mean by understanding here. And so imagine that the brain is this toaster, and an alien came down, and she wanted to understand how it works. There's two different approaches that she could take. One is a more structural, or it's the tradition of the anatomist, or maybe in the modern reincarnation, a more molecular approach, where you're trying to understand the structure, how this thing is built. And the other one is more the physiologist approach, where you're trying to build a faithful model at the functional level. Like basically, what I used to do when I was a graduate student, you can make toast with an iron board if you don't want to buy a toaster. And it works very well. And actually, you can even do other stuff. You can melt cheese. It works even better. And electrophysiologists or physiologists, and some of them will still argue that, why do we even care about the structure? If we understand the functionality of the system, and in some ways what I've described in the first part, this system identification way is trying to do that, we can understand something at the algorithmic level and maybe even implement it in silico and build computer visions that mimic the brain's algorithm, but without really understanding how biology implements it. And I think that's a fair argument. Now, the problem is that if the toaster breaks down, which is our brain, and we need to fix it, if we do not understand it at the structural or molecular level, then it's going to be hopeless trying to fix it. And at least us in the United States, we get funding from NIH, and its main mission is not just to understand how the brain works, but to try and fix it. So it's very important to get to the structural level. And to do this, my lab, we study, we have 3 levels of analysis, or 2 main levels. We study the cell types of the brain, of the cortex in this case, at the transcriptomic level, morphological, electrophysiological level. Then we try and understand the principles of connectivity, and I think Tony will talk a lot more about this tomorrow, and also the function. And in order to relate structure to function, we need data where we have both type of measurements simultaneously. And these are very hard to get this data, because if you want to, let's say if you want to have a neuron and you want to know its input, its morphology, and then you also want to know its electrophysiological properties, there's not that, these are really difficult data to get. And we were lucky enough that a few years ago, Jacob Vogelstein and David Markovich started this program funded by IARPA, which is like a DARPA-like mechanism in the United States called Microns, whose goal was to do functional connectomics at scale. And the idea here is that in this one mouse, we would get data and image about 100,000 neurons, about 1 millimeter cube, and then have the full connectome reconstructed. And after many, many years, this program lasted for 5 years, and a lot of work, we finally collected this data, and I'll show you the data, and we publicly released them like 2 months ago. So the first experiment, the data were collected, were collected by Paul Fahis, Tolias, Jacob, and Jokun in my lab, where we imaged in several mice 1 millimeter cube in each mouse, and it included V1 and extra-striate visual areas, about 100,000 neurons per mouse. Then we shipped these mice to the Allen Institute in Seattle, where Nuno Tacosta and Clay Reed did electron microscopy in one of these mice, because it took months, almost a year, to completely image, create the pipeline and image this mouse. And then the data were shipped to Sebastian Sang, who, using some methods similar to the ones that were showed earlier this morning, reconstructed the connectome. And then we align this data. So in this mouse, we have both the and we use these system identification methods, including to functionally characterize these neurons that I described earlier. So in this one mouse now, we have pretty much all layers down to layer 5. We have functional characterization of about 70 to 80,000 neurons in this particular mouse. And then we have about 1 billion synapses. And this is just a little chunk of a pilot mouse, but these are data rendered and played in real time without showing the connections. This was a small slide for some pilot study that we had to do in phase 1, but it shows you like how rich this data is, because now we have morphology, we have the connectivity, and we have the electrophysiology. And as I said, these data are publicly available to anybody to download and try and analyze them. So for the remainder of the talk, I want to switch gears. I've shown you how deep learning and large data science can be combined together in this modern era we live in to hopefully try and go a step further and create some systematic ways to study and dissect function and structure and combine them together. But this meeting is also trying to go the other way around. By understanding the brain, can we build smarter machines, like advanced AI itself? We all say, that deep learning draws inspiration from neuroscience, but can we directly use neural data to advance AI? And in fact, the main goal of the Microns project was trying to figure out, can this neuroscience data help us advance AI? And we all know that, deep learning is called the fourth industrial revolution. It's overtaking many areas of industry, and it's being used in science. And throughout this meeting, including in my talk, we describe methods using deep learning. But there's also a lot of limitations. And in fact, Matthias Bethke, who is here, who was part of our Microns team, has published some wonderful work exposing some of these limitations, among others. And I will first describe some of these limitations. And the two of them that my lab is focused on with my collaborators is this thing called robust vision and lifelong learning. And in robust vision, one of the famous sort of examples of robust vision is this so-called adversarial attack. So if you train a neural network to discriminate, let's say, cats from dogs, from tomatoes, and so on, it can do an amazing job, and it can reach almost human-level performance on new novel images. But if you take one of these images, and this was known since the early days of deep learning, and you can perturb it in a particular way, and there's many different ways how to do it, and you can fool this network. You can even do what's called targeted attack, and now a frog, by changing a few pixels here and there that are imperceivable to a human, you can never see them, this network now is 100% convinced that this is a crocodile, or you can change it to thinking it's me. Okay, so this it's a security concern potentially, but even more importantly, it exposes the qualitative difference between deep learning and the way our brain works. I actually think this is, and some people even went as far and saying that basically deep learning is like a fancy lookup table. And because it's trained on so much data, it finds relationships in this data that help it predict, but when you try and really understand it deeper how it works, it exposes this limitation. Another one, and this is a very nice review if you're interested, written by Matthias and his collaborators. It shows that it's not just this adversarial stuff, but there's many cases. And one of the most famous one is this cow here, where if you take a cow and you put it on the ocean, these neural networks don't recognize it anymore as a cow. So there are learning features in images that have little to do or not as much to do with the way we perceive the world. So this is a fundamental limitation of deep learning. And as far as I know, in the last 10 years, despite all this excitement and revolution, there's been progress in trying to expose the limitation, but not as much as trying to fix it. I mean, when people are trying, it's just there's been little progress. And at the heart of this problem is generalization. These things generalize very well when the training and testing distributions are very similar, but now if you perturb it, like you put the cow with the background of the ocean, it cannot do it anymore. And we wrote a review about this 2 years ago with Matthias and others, and Fabian and Zach, and a bunch of people, on how kind of like neuroscience, you can use neuroscience data, and I think Tony will talk more about that too tomorrow, to learn the brain's inductive bias. And we kind of categorize this at 3 levels for this review. You can learn this at the computational level, where you try and change the objective function of many objective functions of what you're trying to do. You're not just doing object recognition, but trying to learn other things. You can do it at the representational level, which is the level that I described in the first part of the talk. Or you can try and do it at the implementation level, where you're trying to understand, and again, this is more related maybe to the genes, what is the wiring diagram? What is the architectural constraints that these networks have that enable it to do better generalization? Now I'll describe you some work that we did that we call neural regularization with Jelly, which is a joint postdoc between my lab and Zach's lab, where we try to do this at the representational level. And the idea here was that if we measure the responses of neurons, let's say thousands of neurons from the brain to thousands of images, we can learn a representational manifold, which is the relationship between different images with respect, in the activity space. So for instance, here is like 2 cats, a dog, and you can think of this as the high dimensional manifold. And the idea is that when you train a neural network, you have an objective, which is to try and, let's say, do it to classify cats and dogs. But at the same time, we wanted to regularize it. We wanted to try and co-train it, if you want, to try and mimic the brain's representations. And here is just, you can read the paper was published in NeurIPS 2 years ago. But the idea is that we computed these similarity metrics, in fact, using the same model that I showed you earlier, this predictive model that we used to find the MEIs. That same model, we used it where we passed 100,000 images through it, and then computed this 100,000 image manifold. And then during training, instead of just having 1 loss function, we combined the 2 loss functions together and try to regularize it. So the representation of these hidden layers, we're trying to put the cats and dogs or the images more similar to the brain. And what we found is that these networks, when they were regularized, and here is shown in this red, they started being more robust when we were testing them with noisy images that were never used during training. And we've shown this with many different ways. We have another paper with Fabian Sind that's coming out in NeurIPS this year with monkey data doing something very similar from V4. But the idea is that these networks, when you co-train them with neural data, they tend to perform a little bit better. Now, they don't, they're nowhere close to human level performance, but they improve their performance from the baseline. And with Matthias also, we've done this with adversarial attack, and you can see here one of the very, Matthias's team developed a very aggressive attack called the boundary attack. And when you use that, every single network that fails that was ever published, when we take these baseline networks and we regularize them, they also show some improvement in performance. And this is ongoing work, but we recently found out that the reason that these networks learn, there's a quite simple reason in a way, is that when you train a neural network, if you think of it in terms of the power spectrum of the images, standard neural networks rely more on high frequency information, whereas these perturbed networks, they are more robust they rely more on low frequencies. And when you look at other methods that people have used, not related to neuroscience, they also follow the same principle. So just to, this is my last slide. Guido, one of the reasons that we think the brains are different, and a lot of people think that, is that brains use more generative models than discriminative models. And if you're interested to learn more about this, and about generative replay, you should go to Guido's poster this afternoon. So to conclude, I think this is a very interesting era where we have technologies to record a lot of data. We have tools that we can build these models, despite their limitations, that then can give us insights. And then we are sort of in a new era, I think. It's a starting field where we're starting to see some early evidence where neural data directly don't just inspire AI, but maybe can inform AI. Thank you.